y después de grabar lo pongo todos los videos a la página para que ustedes puedan volver a verlos. Okay, so today we have two topics I'm going to go through today. One is about how to construct so-called a weight matrix, how to characterize binding motifs in sequences, and then the other part after the break is about sequence profiles, a very powerful tool to characterize protein families, protein sequences, and identify in a very quick and intelligent way where do you expect to have conserved positions, active sites, and so on. So I think it's a very powerful tool to apply if you do protein science to get a quick and very rough estimate on where in your protein is all the fun happening. So these are the two topics of today. Please, during my presentation, if I go too quickly over something or if things are unclear, please just interrupt me. I think it's better to do it while I'm talking compared to waiting to the very end. So please don't be shy. Please stop me and ask questions along the way. So let's go and start with the first presentation. I have to remember to share screen. Okay, so now I hope you see my screen. So the first topic, as I said, was how to construct a weight matrix. What are they, and how can you visualize in a very powerful way the information content kept in such a weight matrix? So a weight matrix is something that characterizes binding motifs. And in biology, a lot of stuff is happening by small linear motifs describing an interaction surface between different protein components. One is, for instance, a signal peptide in a protein that dictates where in the cell does that given protein end up being located. Another important thing is phosphorylations, for instance, PTMs in protein sequences, which sites in a given protein can be modified by phosphorylated modifications like phosphorylations. And again, the motif that defines if a given site can be modified is defined by a linear motif, as we'll go over today. So these are kind of very important aspects describing a lot of biology. So to know how you construct these motifs gives you a very good tool to learn about the function of protein and things happening in interactions between cells. So the idea is that you have some short peptide protein fragments that each of them share that they do something. They can be phosphorylated, they can be binding to a given receptor. But all of these peptides, they share this common thing that they can do a certain function in the system. And then you want to use those data to characterize the so-called binding motif, meaning if you look at these peptides, do they have something in common at certain locations? And going through all that, we're going to go through today in this lecture, we're going to end up having a sequence motif like showed here on the left, right? So you can see very quickly from looking at the peptides themselves, it's very hard to figure out what are the patterns that are shared between these peptides. Looking at the motif, it becomes very clear that you need to have a proline that is conserved in the middle of the peptide. And then you can have a charged amino acid to the left of the proline, two amino acids to the left, and you have a proline conserved to a lesser degree, free amino acids to the other side, right? Then you have a quick overview. And once you have the overview, you can also almost by the eye scan through the other protein sequences and see, do they have that given motif, and thereby do those other protein sequences or segments from protein sequences, can they execute the function you have been investigating? So how do you come from the peptides on the left onto the binding motif on the right? This is the main topic of today. So the reason why it's so important is that if you understand how to make these weight matrices and characterize a binding motif in these logo plots I showed just before, then you have a good tool moving forward into your understanding of processes in biology. So we're going to go through all of this. We're going to show you how to construct a weight matrix from these peptide input sequences, and there are some tricks of the trade, how to do that and make the motif as accurate as possible. So this is, I think, the first topic of this course where you actually deal with what I would call bioinformatics in a nutshell. So bioinformatics to me is that you have some data and you try to learn the shared pattern in those data, and then you thereby learn the rules saying that all the peptides that bind to my receptor, for instance, they must share these properties. And once you have learned those rules, you can now go out and screen through the proteome of interest and see, can you find peptides that share that property? So we have done that in my research over and over again, and here example is from peptides that binds to a very important immune system receptor called MHC. And that MHC molecule displays peptides to the immune cells, the T cells. So if you want to, for instance, in this case, learn the rules defining which peptide binds to my MHC molecule, you go through the techniques of today, you construct this sequence logo shown here in the middle on the left, and now you have the rules that the peptides that bind to my receptor, they must have an L at the second position, then they must have something pretty arbitrary, and then at the last ninth position, they must have a V or an L. And those rules have now been learned by these simple sequence logos. Now you can go out and screen through the proteome of SARS-CoV, for instance, and find peptides in the SARS coronavirus genome that could bind to that receptor and thereby hopefully make a vaccine that will work and cure people for the next invasion of the next SARS-CoV variant. But this is kind of, in the essence, what we do in bioinformatics. Learn the rules from label the data and then apply those rules to make cures or modify systems to make them behave in a certain manner. So I'm very fond of the immune system, as you might hear from what I've said before. And the immune system is of course, essential. We know that to cope with infections. And one of the important players of the immune system is this MHC molecule shown here in the ER. So what happens in the immune system is that you have proteins being produced in the cell, both healthy, normal proteins doing the cell function, but also if the cell is infected by coronavirus, then some proteins will be coming from the virus origin, and all of these proteins will be digested, into small peptide fragments by the proteasome, and some of these peptides will meet the TAP transporter molecule and go into the ER, where the MHC molecule awaits. And if the peptide have the right amino acid sequence, that means they match this binding motif we have characterized here of the MHC molecule, then they might bind to the MHC molecule and go out to the cell surface, where the immune system T cells will await and scan for peptides shown on the cell surface bound to MHC of foreign origin. So if a cell here is infected with coronavirus, then some peptides out here on the surface bound to MHC will come from proteins from the coronavirus genome, okay? And these peptides can be recognized by these T cells. And if that happens, the T cell will induce apoptosis of the infected cell. It's really an effective way to clear infections. So here we see the T cell is scanning the surface of the cell. And if some of these peptides on the surface are of foreign origin from infections or from tumor cells, something that doesn't look like self, then the cell is killed. And I have a small video, I thought, showing you this. Where is that video? Here it comes. So that here you have a T cell in the lower part binding to an MHC peptide molecule shown on the bigger cell. And since this peptide was from foreign, it was an infected cell presenting on the cell surface a foreign peptide, then this cell was told to undergo apoptosis. So understanding this MHC presentation of peptides is important to understand how the immune system works. Let's just leave it at that for now. Where am I going now? I'm going the wrong way, sorry. So we would like to learn the rules. How do we characterize these binding preferences of a given receptor? And one important concept to quantify this is what is called sequence information. Okay, so we have some peptides here to the right, and we want to from those construct the rules of binding. So sequence information can be formulated in this way. Say we have some rules, we have some data, and from those data we can learn that all the peptides, they have L at P2, right? Nothing else is found. You see here L at P2 all the time, right? Whether if you look at P1, you can have any of those 4 amino acids, A, F, W, or Y, right? So which position, P2 or P1, has the most information? You can reformulate that question by saying, how many questions do I need to ask myself if I come with a new peptide and I know the rules from up here to tell if it's a binder or not? If you look at P1 here, you have to ask 4 questions, right? To have an A, yes, no, an F, yes, no, a W, yes, no, or a Y, yes, no. If you look at P2, you only have to ask 1 question. To have an L, yes, no. So you can much more quickly define binder, non-binder by looking at the information at P2. So P2 has the most information content, right? You can then formulate this in a more mathematical way. You can find the frequency of each amino acid at a given position. Then you can write down the sum over the 20 amino acids here of P times the log of P. That is called the entropy. And then information content is just log of 20 minus S, right? Or this is if you assume that all amino acids in the world have the same background frequency of 5%. In reality, we know that certain amino acids are more frequent compared to others. And if you want to incorporate that, we write the information content in this way, where we take the ratio of the observed frequency in our data divided by the background frequency of that amino acid found in nature, just scanning through UniProt, a big database of proteins, and estimating a background frequency. So looking at these equations, you can see if you have a fully conserved position where you only allow 1 particular amino acids, like we had before here at P2, where only L was allowed. In that case, you get that the frequency of L is 1, and the frequency of all the other 19 amino acids is 0. Plugging in those numbers to these equations up here, you get the entropy being 0 and the information content being log of 20. If you have a highly variable position where all amino acids are tolerated and equal frequency, then each amino acid frequency becomes just 1 over 20. And again, going up to this equation here, the entropy becomes log of 20 and the information content becomes 0. So high information content for low variable for highly conserved positions, and low information content for highly variable positions, right? So now if we have a large data set like this, and all these peptides, they are known to bind to my receptor, can we then apply those rules to extract the information content per position? So we do that by first calculating a matrix of 20 amino acids here, A to V, and the 9 positions. And then we go through these data column by column and calculate the frequency of each amino acid type at each column. So we fill out this matrix, 20 times 9, and each number here is the frequency of that amino acid type at that given column in my alignment. And then we apply the rules from the previous slide to calculate the information content, right? Log 2 of 20 plus the sum of those frequencies times the log of that frequency. Doing that, we get the last column out here, information content. Now we will see that the information content at P2 and at P9 are relatively high, 1 or close to 1, and the other numbers are close to 0, right? So you have information at P2 and at P9, and in the other positions, you have limited information, meaning that you can have any amino acid type you would like to have at that position. So now we can use that information to draw the so-called sequence logo. The logo is just a way to represent what is in this matrix. So we take the last column, the information column, and that defines the height of the given bar at each position. So the height at P2 is something like 0.4, right, or 0.6 here, and the height at P9 would be close to 1. So from the bottom up to the top, you have a height of close to 1. And then the height of each amino acid is the relative frequency at that position. So here we have L having a frequency at P2 of 62%. So L occupies 62% of the total height of the bar. Okay, and then finally, we have characters shown on the positive and negative y-axis. The one on the positive y-axis are the ones where this value from the frequency here is bigger than what we expect in nature by just by chance. So this value of Q from my previous slide here, this value of Q, the background frequency for an amino acid type found in a big database of protein sequences. So if P is bigger than Q, right, then this ratio is bigger than 1, and the log is positive. Then you plot these amino acids on the positive y-axis. If the ratio is less than 1, then you plot them on the negative y-axis. So these guys are depleted, the one down here, and these guys are enriched. Okay, and now you have the sequence logo. And you can now see again that this MHC molecule, the AO201, prefers peptide with those characteristics, an L at the second position, and a V or an L at the last position. So you know what to look for when you go into your corona proteome and want to find peptides that can bind to this receptor and hopefully give an immune response and help you cope with the infection. So you want to make a vaccine based on these peptides. So this is kind of looking at the protein structure of this MHC molecule with the bound peptide. You will see that these information content rich position P2 and P9 are the ones where the binding takes place between the peptide and the MHC. You will see here that there is here, this is the second position. It is really hidden deep down into the binding cliff. The side chains are pointing into the binding cliff. And likewise, over here to the left, we have the C terminal, the 9th position, and again, the side chain is pointing deep into the cliff, right? So again, you have this correspondence that the high information positions are the ones where the interaction is strong between the peptide and the receptor. So now this is all me talking, and as always, I think me talking a lot is not a very good idea. So I would like to ask you to do the first exercise in the course. Let me just go back to the slide here. Here, so here we have the first exercise with logos, right? If you open that link, you get to a page like this. Okay, and then your exercise is to go through this document and fill out this table, go through the questions, fill out this table, and then try to draw. We cannot have the whiteboard here, so we're just going to discuss this when we go back and meet again in 10 minutes. But you don't have to do it for all the positions, right? I just want to go back and show you one example before we start. So here, if you say you have to fill out this table I showed on the second page of the handout, and if I take position 0 and work on that, right, which is the one in the middle, then this position is fully conserved. All 35 sequences have a G there, right? So it's trivial for me to do that. I get 0, 0, 0, and 35 Gs. Then I convert those counts into frequencies, by dividing each count with the sum of the counts. So here I get 0, 0, 0, and 1. It's pretty easy. I have to fill out the next table, which is, again, what is the entropy and information content. So again, entropy, here you have a fully conserved position, and you go back to the equations for entropy. They were here, right? Entropy here. So you have something that is fully conserved. 19 of the so 3 of the p's, sorry, are 0, right? So those terms with those nucleotide bases, the p times log of p when p is 0 is 0, and log of 1 for the fully conserved G, log of 1 is also 0. So all 4 terms in that summation gives 0, right? So for a fully conserved position, the entropy is 0, and now the information becomes 2. And now you're done, right? So try to do that. Pick another position. Don't pick position 0, but I've already done that for you. Pick another position of the ones shown in the handout in orange here on top. So one of these orange positions here from 6 to 5, and go through filling out the answers to that particular column in this table, that particular column in this table, and so on. Okay, and then we will meet again in 10 minutes. I would suggest that I make some breakout rooms so you don't do this on your own. And I'll just make them make breakout rooms here. So I'll just make 10 breakout rooms, so you can just I assign you to them automatically. You can just move around if you want to stay with your colleagues, but now they're all open. So please go to a breakout room, and then we meet again in 10 minutes. That means 14. Is that correct? 10 minutes. Yeah, 14 minutes to. No, I cannot get it. Yeah, 16 minutes to 11. Is that true in Argentina? I don't know where we are in the world. So at least we meet in 10 minutes. So please, I'll close the rooms in 10 minutes and try to get this done. And if you have questions, of course, in the chat, you can let me know. So, and I will share screen here. So here I have my answers. So please, I think it is not complicated. You can do some counting wrong, but the critical thing is that you count at each position the number of different base pairs you find. And then below here in the columns with the frequencies, you divide those numbers by the sum of those, which is always adding up to 35, because you have 35 sequences in your alignment. And then you get frequencies out here. Then you plug those numbers in to this equation 1, to get the entropy. And then this is the entropy is always done with respect to logarithm with base 2, and the relationship to go from the logarithm on your calculator or in Google or whatever you use is to take the log in whatever base, the log of natural logarithm or the log 10, and then divide that by the log of 2. That gives you the log with base 2, right? This is the only little trick there is. And then once you have found the entropy, you get the information content by taking 2 minus the entropy. So doing all that, you get these numbers here, right? So you have these highly conserved positions, 0 and 1, where you have no entropy and the maximum information content. Then you have variable degrees of information content at the other positions. Do anyone know why you have this number 2 here in the beginning of the equation 2? In the slides, I had the log of 20 at that position. Anyone who wants to comment on that? Not really. So log of 20 in the slides, I was describing the amino acid alphabet, right? Where you have 20 different amino acids. Here we are talking about DNA nucleotides. We have 4 different nucleotides. So it should be the log of 4, and the log 2 of 4 is just 2. Right? So this is kind of the log in base 2 of 4 gives you 2 here. Okay, and then just to close it up, you were supposed to now manually draw on the whiteboard the sequence logo based on these information content numbers, and then the frequency values you have for each of the base pairs up here. We cannot do that because we are online, but you can show it by going into this web logo web page and upload your sequences, and doing it, you get this logo back here. Okay, so now you see here, now the numbers are changed because that's how they do it. Here we had that the position in the middle is called 0 and 1, and these are called 6, 7, 8, and 9. Now this is called starting from 1 up to 10. But you see here the conserved positions in the middle, G and T, and then the variable degree of information content on the flanking regions. And this logo here, for instance, at the G at P4, corresponding to this position 9 in the alignment, has the height of G is the biggest, followed by A, T, right? And then if you go back to your frequency table up here, you see here that G is the highest with a height of 74% of the total height. Right? So this G here occupies 74% of the total information content height, right? And then the A is the second, because the A has 11%, then T has 8%, and C has the last 5%. So you color them according to the frequency, and you sort them from high to low, most frequent on top. Okay. I hope so. Otherwise, yeah, let me know in the chat, or however you want to talk to me. I hope it's fine. So let's go back now and continue with the presentation. Have a little bit more to go before we have a short break. I think we were here. So this is how you make a sequence logo. If you have a lot of data, but very often you have to kind of save some money, or it's complicated to get access to large volume of data. So very often you want to find a way to make a sequence logo, even though your number of peptides that characterize the binding preference is limited. Okay, so there are some very simple tricks to do that you can apply to boost the performance in case you have limited data. And it works for these receptors we talk about today, but it works also later on in the course, where you talk about motifs for disorder and other structural domains in proteins. These techniques you hear about here are valuable across the board, right? So here we have a small set of peptides, right? Only 10 peptides are available to us, and we want to apply the techniques of making a sequence logo from these 10 peptides only. So what can we learn? Looking at the data themselves, we see here that at P1, the first column, we have 6 out of 10 peptides having an alanine, right? That could suggest to us that alanine is very, very important, because more than half of the peptides share that amino acid at that position. We also see at the last column here, we never observe an I. We observe T and V and M and so on, but never an isoleucine. Does it mean that if you have a peptide with an I, it will never bind? Maybe not. Making so strict rules from 10 peptides is maybe too harsh, right? So it's very hard from small observations to make very good rules. The most simple rule you can make is a regular expression shown here, where you just take what you have seen in your data is allowed, right? So you have seen an A, a G, a T, and a K, A, G, T, and a K. So those are allowed. Everything else is disallowed. At P2, the same thing. You have C and M, I, and an L. So these are the allowed amino acids. Everything else is disallowed. This is the most simple way of characterizing a binding motif. What you have seen is allowed. What you haven't seen is disallowed. And it's clearly not very good. If you only have 10 peptides, your rules become very, very, very limited. Looking at the data we had in my previous slide, the one with all the peptides binding to my MHC molecule, these rules constructed from these 10 peptides will only allow 11 of the 212 peptides on that slide to be recovered as binder. Right? So you really discard the vast majority of known binders by these very, very simple rules. So we're going to do something more sophisticated. Okay, sorry. And another way to show that this really doesn't work very well, if you have these three peptides here shown on this slide, with the according where you have measured the binding affinity to my receptor. Here, a low value means strong binder, and a higher value means a weaker binder. Then applying the rules from the regular expression on top here, only the first peptide will not match, right? Because there's not an R at P1 here, so it became a non-binder. Likewise here, this S is not part of P7, so it won't bind, right? So these two peptides will be deemed non-binding. Only the third peptide will bind according to those rules, but that's the weakest binder of the three, right? So again, these rules are clearly too stringent if you make such a regular expression based on just 10 peptides. We can make it a little bit more sophisticated by saying we just don't make a regular expression. We weight the amino acids based on the frequency in the alignment. Okay, so since you have here 6 alanines at the first position, you would assume that alanine is more important than G, and it's more important than T and K, and the other amino acids are not important at all because you haven't seen them. So that helps a little bit because now you get can rank peptides, not just binder, non-binder, but you can rank them how well they bind to a degree, how frequent are the amino acids you see. So here the alanine here will weight higher compared to the G here, it will weight lower, right? So that's kind of a way to move forward. But again, you are struggling with very few data points to make that statistics. So it might not be the best approach. So what you really want to do is do something novel. Before doing that, just look at what you do if you take those data and go through what we did on the handout in the exercise just before and try to calculate the frequency of each amino acid at each position, get the information content, and you can draw a logo like that. This logo doesn't look like anything like the logo we had in the beginning with these two very sharp position 2 and 9 where the anchor were defining the binding of our peptides to my receptor. So we have to do something new to move beyond that. One first thing to do that is very, very powerful is to look into sequence weighting. So in these 10 peptides, we clearly see that there's a group of peptides that look very much alike. Had you gone out in nature and sampled 10 peptides, it's very, very unlikely you would have found five peptides so similar by a random sampling of what happens in nature. So this clearly suggests that there is a bias in the data, that these data were made in some artificial way that gave a high repetition towards these peptides. So we want to downweight that. So we're going to reflect in our data what we will find in nature. That's called sequence weighting. So we take those 5 peptides and we say they don't count as 5 instances, they count as 1, right? So that each of these alanines here count as 1 divided by 5. Each of those L counts as 1 divided by 5. This M counts not as an M, but as an M divided by 5. Right? Doing this, you remove that bias towards these peptides that is an artifact from how the data were generated and do not reflect what you find in nature. Doing that, we get a little bit better sequence logo. Right now, if you go back here to this slide, you saw alanine all over the place being the most important amino acid because we had these peptides in the beginning of our list with all the alanines. Now, by doing this trick, these alanines have been removed. Right now, they are further down in the motif, and we see something more refined about which amino acids are important. But again, we only see the amino acids that we have in our data. So if we haven't observed a peptide with an I at the last column, we never see an I over here, right, in the last column. So still we're limited by the very small number of observations we have to make meaningful estimates of these amino acid frequencies. This is the last thing we want to resolve. So looking at the last column here, we haven't seen an I ever, but we see something that looks like an I from a side chain perspective. You know, amino acids have the side chain, and some amino acids, even though they have different side chain, the side chains are very alike to each other. So isoleucine and leucine here are very similar. It's just a very, very minute change between those two that distinguish them. So if you can have a peptide with an L at the last position, it's very likely that if you replace that L with an I, the peptide will still bind to my receptor. So how do we quantify that? How do we quantify the likelihood of, if I replace something I have here at the last position with an I, what's the likelihood of the peptide still binding? We can use the Blossom matrix for that. So you have seen the Blossom matrix in this way in the course when you talked about alignment of protein sequences. This is a symmetric matrix giving the penalty for mismatching in alignment. In a mathematical sense, this matrix is found in this way. If you look at, you can rewrite this matrix by taking this Pij divided by I. It becomes the conditional probability of, if I observe amino acid I in one given protein sequence, the likelihood of that being replaced to J and still maintaining the function of the protein. And this matrix here, the P of J given I, is shown up here. Right? So there's a map between these two matrices. This is the one we want to use today. This is the one you used earlier on when you talked about alignments. You can see you can go from one to the other. If you want to look at the score for A versus A in this matrix here to the right, the score is 0.29. Right? So this is what you have here, P of J given I. Now, if you divide that by the background frequency of alanine found in UniProt, which is 0.074, that ratio, take the log 2 of that and multiply that by 2, you get 3.9. And again, the value over here in the Blossom scoring matrix you've used earlier is 4. Same thing. Likewise, if you go for the value for R given A, right? So this is the one here next to it, 0.03, divided by the background frequency in nature of R, which is 0.052. Again, that ratio, take the log of 2 and multiply by 2, you get minus 1.6. And again, if you round it off, you get minus 1. So this is a direct link between those two. So now what does this matrix mean? It means that if you have something in your observation, you can estimate a likelihood for that observation being changed to any other amino acid, right? We know that cysteines like to be highly conserved in proteins, so you have a low chance of changing it, meaning that the cysteine is very high. But isoleucine, for instance, looks very much, as I said before, like leucine and like valine. So you have a very high chance, if you can observe an isoleucine, you can also observe an L or a V, right? So these are interchangeable, and you maintain the binding properties or the protein structure to a very high degree by exchanging those 3 amino acid types. Now we can apply that to estimate a frequency for isoleucine. say that we haven't seen it in our data, but we have observed the valine, right? We go up to our table, say we have observed valine here. We go in and we find isoleucine 0.16. And now we have estimated a frequency, the likelihood of having observed a valine in our data. We can replace that with isoleucine and maintain the binding properties. We now write it down in a more formal way. Here we can say the pseudo frequency at a given position of a given amino acid type is the sum over all the observed amino acids in that given position, multiplied by the blossom, the likelihood of changing the observed amino acid into the amino acid we're looking for, B, right? So again, going back to our data here, looking at the last column here up to the right, we want to estimate the pseudo frequency for isoleucine, right? We go through the summation. Summation is over all 20 amino acids. So we take the observed frequency of alanine, for instance. So the first term here is alanine. The observed frequency of alanine is 0 because there's no A's in the last column. So that term doesn't contribute. So we end up having only these terms in our summation where we have that amino acid type at the last column. Okay. So here we have an M. The frequency of M is 2 in 10. So we have 0.2 multiplied by the blossom frequency matrix of changing an M to an I. Going on, we have an R. We have 1 R out of 10 times the blossom score for changing an R to an I. And so on, we sum up over all the observed amino acids, go back to our matrix here, find the numbers. And it's very important when you find these numbers here that you read the matrix right. If you go back to this matrix here, you might realize that the matrix is asymmetrical. So the value for N and A is 0.04, but the value for A and N is 0.03. So you have to read this matrix in the right way to get this calculation done right. So what you have in your data is on this end, this axis, and what you're looking for to replace into is on the other dimension. So here you have what you have is the M, and what you're looking for is the I. What you have is the M, and what you're looking for is the I. So it's here 0.1, right? 0.1. And you go on doing that for all the amino acids, and you add up and you get 0.94. Right, so now you have the so-called pseudo frequency value here. You have the observed frequency for all the amino acids in the given column, and you want to find a way to combine them. You take this weighted average, alpha times the observed frequency, plus beta times the pseudo frequency, divided by alpha plus beta. Okay, and what is alpha? Alpha is the effective number of sequences, which is in essence just the number of sequences minus 1. And beta is the so-called weight on prior or weight on pseudo count. It's an empirical number that you estimate once and for all and use that moving forward. And that number is in general somewhere between 50 and 200, right? And I will show you in a minute a way to estimate what should that value be. So now you have found this equation, you have the values for alpha and beta. So what does it mean? Say you have an alignment where you have a lot of sequences. You're lucky and someone has made thousands and thousands of sequences to characterize your interaction to your receptor. So alpha is maybe 10,000, beta is 50, always 50, between 50 and 200, say 100. So you have 2,000 here and 100 there, meaning that alpha is so much bigger than beta. You just ignore the term with beta, so you get that P becomes equal to F, right? So if you have a large data set, you don't really care about extrapolating and using pseudo counts because the data themselves tells you everything, right? And you can just use the raw counts to estimate your frequencies. If alpha is small, like here, you only have 10 peptides, so alpha is 9, beta is still 100, right? So now this term becomes small and P becomes equal to G. And then it becomes very important to include these pseudo counts, right? So it's kind of the idea that if you have a large data set, you don't care about pseudo counts, the raw observations is sufficient, If you have low observations, pseudo counts are very important because you have only seen a small proportion of the total space of binding peptides. So now applying that trick to our data up here, still only the same 10 peptides as before. We do the sequence weighting, we do the pseudo count, and we get this logo back. It still doesn't look very good, right? But now we have at least recovered that P2 is the most important. P9 is still doesn't look very well. We have the right amino acid types, but the information content is very low. But more importantly, we see here, for instance, at P9, the 5th most frequent amino acid here is the I, right? Even though we have never seen the I in the data, using these pseudo count frequencies from here, we get a non-zero value for I, and that means that this becomes the 5th most likely amino acid at that position, right? And then the last trick we can do is that, say you have done this exercise many, many times for many different receptors, and you always find that P2 and P9 are the most important amino positions in the peptide with the most information content. So maybe you can just cheat and say, maybe that rule also works for my new receptor. So I just increase by multiplying by a factor of 2 the information content at P2 and P9. So now you have arrived at this final result, okay? And this result is derived from just 10 peptides. And now we show here below the motive found if you worked with all the 212 peptides from my first slide. So you really come a long way, right? You see the amino acid preference is close to perfect. LMIV, LIMV, and over here, same thing. We have the M sticking in. Otherwise, we are close to being perfect in our ranking of the amino acid types. So just from 10 peptides, applying these simple techniques of sequence weighting and pseudo count, we have been able to extract a highly accurate binding motive. And then the last thing we want to do before we are done is to now we have found these frequency values here for all our 20 amino acids at each position in the peptide. How can we interpret that? What does it mean that here at P2, the value for M is 6% and the value for V is 10%? Does it mean that V is more important for binding compared to M? Maybe not, because we know that in nature, all amino acids are not found with equal frequency, right? Looking at nature, we find that on average, we only have less than 2.5% M's, and we have 7.3% V, right? So this number here, 10%, is just barely above what you expect by chance, whereas the M value here is more than twice the value you expect by chance. Somehow we have to recalibrate the frequencies we observe in our data to what we expect to find by just looking at chance in nature. And we do that by dividing the frequency values here by the background frequency found in nature. So again, going back to the slide before, we take this value of M divided by 0.025, take this value of V and divide that by 0.073, right? So then this ratio here becomes more than 2, and this ratio over here becomes just a bit over 1, right? Meaning that the ratio M of 6% is actually more enriched compared to what you would expect by random compared to V. And now we take in the end the log to make the values positive. So if the P is bigger than Q, right, then the ratio becomes bigger than 1. The log is positive. If P is less than Q, then the ratio becomes less than 1, and the log becomes negative. Now you can directly read off which amino acids are enriched and in favor for binding, and which are in disfavor for binding, the ones with negative values. Okay. Now we also see here that the M has a value of plus 3.1, and the V a value just barely above 0. And then the last thing to do is now we have made, this is the final weight matrix that characterizes the binding motive of our receptor. Now I want to use that matrix to go out in nature and scan through a genome to find other peptides that also could bind to my receptor. And how do we do that? We take simply the peptide, and then we score each amino acid against the matrix. So we take the first amino acid, find the first row, and take the amino acid type R 0.4. The next amino acid is an L at the second position, scores 5.1. And the next L at P3 down here scores 0.3, and so on. So we keep going. We're adding up all those numbers, and then the final score is assigned to each peptide. And the higher the score, the more likely it is that this peptide binds to my receptor. Right? So in this case, the second peptide, GLLG, is the one with the highest score, the one with the highest likelihood of binding to my receptor. And the last one here has a lower score and therefore the least likely to bind. And again, comparing to what we did in the beginning, where we had measured binding affinity values, where we found that this GLLG peptide was the strongest binder with the lowest value here. Now the prediction scores align perfectly to what we observe, right? And again, this is arrived by using only the simple techniques of sequence weighting and pseudo count. So I know we are all maybe tired, but I think we should just do the next exercise before we have the break. So I'll just stop sharing here and ask you to go back to the course program here. And then there is the second exercise here, where we're going to go through all of this we have done today with pseudo counts, right? So again, here we have an alignment here with 6 peptides. We're going to focus, this is very important, listen carefully. We're going to focus only on the first column, right? So only on the E, E, Q, E, 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 right? So the rest is just to make the problem look like biology, but you only have to focus at the first column. So now you pick an amino acid from this list here. I'm going to do alanine.
Morten.
Yes.
No estás compartiendo pantalla, perdón.
Ay, gracias, perdón. ¿Desde cuándo?
No, de recién.
Ok, pensé que era todo el tiempo. ¿Dónde fue mi...? Acá. Okay. Sorry, guys. Thanks. Okay, so we try again. So this is the exercise, okay, that you have to go through this and answer the questions. You have to focus only at the first column here, the EEQEEE column. The rest is just to make it look like biology. And then you have to pick an amino acid. Right? Pick just one amino acid and then fill out the observed frequency, the pseudo frequency, the combined frequency, and the weight matrix value for that given amino acid. I'm going to go through alanine just now, so don't pick alanine. So when we're done here and I assign you to the breakout rooms, pick an amino acid and go through this exercise and fill out one amino acid only. Okay? And then we meet again in a short while. But let me just go back and show you how it would work for alanine. Stop sharing. So for alanine, if you know that the frequency for alanine is 0, right? In the data, there's no alanine, there's only E's and Q's, right? So the frequency of alanine is 0, the observed frequency. Am I sharing now? Do you see my screen?
Sí.
Yeah, gracias. Okay. So this is 0 here. The pseudo frequency for alanine you get by applying the equation, right? Summing over all amino acids, the observed frequency multiplied by the blossom frequency for changing the amino acid into alanine. In this case, only 2 amino acids are observed. So even though the sum contains 20 terms, only 2 of them have non-zero values for F, which is F of E and F of Q. okay? So we get f of E, which is 5 and 6. We get f of Q, which is 1 and 6. And then we get the Blossom matrices by going into the Blossom scoring matrix and looking up, starting in the column, in the row for E, finding A, starting in the row for Q and finding A, right? So always meant to start, find the row here from what you have observed, and the column from what you're looking for, alanine in this case. Now we get these numbers 0.06. Now the final frequency is the combination of the observed and the pseudo frequency with alpha times the observed plus beta times the pseudo frequency. In this case, I have 6 sequences. So the alpha is 6 minus 1, it is 5. And here I've just said that beta should be 5 to make the calculations easier. So here we take 5 for beta. I know I said that beta was most often 100, but here just to make it easy, we just use 5. So then we get 5 divided by 10, right? So we get altogether 0.03 for the observed frequency. And now we want to have the weight matrix value, which is this value up here, the P, divided by the background frequency for alanine. It is again given in the PDF of the exercise, 0.074. So this is the ratio, the log of that divided by log of 2. 2 times that gives you minus 2.61. Right? So now you have all the 4 numbers and you're done. So now your exercise is just to pick another amino acid, do the exactly same. Okay? I'm going to make the breakout rooms just now.
Marten, una pregunta.
Sí.
¿Puedes? Ahí cuando vos calculás, ¿no puedes volver a la pantallita que tenías
Sí,
recién? Por favor.
Sí.
Ah, sí,
No estoy compartiendo. Un momentito. Tanto se fue todo. Okay.
Ahí va. Cuando vos haces el valor de G, siempre, o sea, tenés que hacer la sustitución por los aminoácidos que están presentes. En este caso vos dijiste que eran 2.
Sí.
Ok.
Sí,
Yo pensé que lo tenías que hacer por todos, por eso.
sí, hay que hacerlo por todos, pero los demás tienen valor F0, ¿no?
Claro.
La expresión tiene los 20 términos, ¿no? Uno por cada aminoácido.
Sí.
Pero si los F son 0, multiplicas el Blossom score por 0, pues no vale la pena hacerlo. Lo puedes escribir, pero no aporta nada porque el F es 0.
Ok, listo entonces. Dale, gracias.
Marten,
Sí.
para el cálculo de P, como son pocas frecuencias, son 6 frecuencias, y vos dijiste que beta va de 50 a 200, ¿no podríamos
Sí.
decir que el término de alfa se puede despreciar frente al de beta en vez de ponerlo como...
Sí, en la vida real, en este caso, el parte acá con alfa y el F va a ser muy pobre siempre, ¿no? Porque si beta es 100, 5 por alfa y 100 por beta, el beta lleva todo el peso. Pero en este caso lo puse beta a 5 para que sea más, como para que contribuyan lo mismo en términos relativos, ¿no? No es un caso real, es más para este caso, para que sea más, que el alfa aporta algo a veces.
Ok, entonces tomamos el beta como 5.
Sí.
Ok. Gracias.
Bueno, si ahora no sé cómo hago eso, si vuelvo a abrir todos los rooms como antes, mismo lugar que siempre, y se van allá y nos encontramos de vuelta en 35, ¿no? En 2 minutos, creo que es suficiente.
Ok.
Perfecto. and then share my screen. So these are my results. I hope you agree. If not, and if you want to discuss things, stay on. We have a break in 5 minutes to get a bio break, as I think it's called. So you can maybe just hang back there. We can discuss if you have particular questions. I also will upload the answers both to this exercise and the one from before to the course program together with the videos recorded today. So any questions here? No? Fantastic. So again, just to kind of appreciate that you look at the data and you get, of course, positive weight matrix values for the Q and the E, as you would expect, because those are the ones you have seen in the data, right? But for instance, the value for D, so D and E, they are kind of similar in side chain structure. So that's still scores negative, but it scores just barely negative, right? So that is kind of the third highest score you get, because you have the pseudo count for D, which is substantial here, right? Same thing for K, you have a substantial pseudo count, and that's why you get a negative, but not so negative values. Anyway, so now we are done, and I just want to recap a few things before we close. That I mentioned very briefly the difference between the Shannon information content, this one we have here in the below the matrix, or the so-called Kullback-Leibler, where you take the ratio in the P here, you divide by the background frequency of the amino acid type. And if you rewrite the Kullback-Leibler equation by saying that the log of a ratio is the same as the difference between the numerator and the log of denominator, then you can say here that if all these background frequencies are 5%, so you assume an equal frequency for all amino acid types, then you can take this Q here out of the summation because it's just a constant, and then you get back the Shannon information content. So the Kullback-Leibler reduces to Shannon if all these Qs here have the same value for all amino acids, a value of 0.05, right? It's kind of nice to know that these Shannon is a special case of Kullback-Leibler where all the amino acid background frequencies are the same. So now we are kind of done. We have seen this. You can make these sequence logos. There's a tool available called Sec2Logo. You can try it out and upload your data, and you get back sequence logos incorporating this sequence weighting we discussed and the pseudo count that we just worked on now in the handout. So I wanted to say just how do you estimate this value of beta? I mentioned that beta had a value between 50 and 200. How do you arrive at that number? It's done by kind of constructing a weight matrix on some real data, in this case, our 10 peptides, applying the techniques you have been going through today, and then you want to evaluate how well does this matrix you have derived predict some data where you have measured the actual binding value, right? And here we are lucky that we have a set of 528 peptides where a colleague of mine has measured in an in vitro assay the binding strength of that peptide to my MHC complex. So we can kind of make a prediction using our matrix by summing up the scores of each amino acid to the matrix as I described before, and then we can see how well does the prediction score align with what was measured in the wet lab. Hopefully get a very good alignment, so you get kind of a scatter plot where you can see a tendency, a high prediction score corresponds to a strong binding value, and a low prediction score here corresponds to a poor binder, right? You can quantify how well these points they associate by, for instance, the Pearson's correlation coefficient. And in this case, you get 0.62, which is not perfect, but also clearly not random. So now we can try to do this exercise and say we can now make a weight matrix and changing the value of beta, the pseudo count, we use to construct the combination of the observed frequency and the pseudo count frequency, right? And we can then make a matrix for a value of beta equal to 0, meaning that we only include the raw F frequencies in the weight matrix construction. And doing this, we get a performance that is pretty bad, 0.48 in terms of this correlation coefficient between the predicted and measured values. If now we increase the beta, we see a general increase in performance. So having a value of beta that is non-zero helps us to extrapolate from these few 10 peptides to the whole peptide space, which is good. And you will see in general that this curve increases, then it reaches a plateau. And either it just stays at this plateau or it starts to decrease again, right? And this optimal value is almost always in whatever you work with of data in this range between 100, between a 50 and 200 in this regime. And this is why I say that beta always has this value of between 50 and 200. That turns out to be the case. In my career, whenever I work with data, I always find that a value of beta in this range is working. So I never do this check anymore. I just always apply beta to be 100, and that's it. So now we're done. We have shown that you can create a weight matrix, a sequence logo from very small number of observations. In this case, only 10 peptides, of which 5 of them were highly redundant, right? But even doing that, just using the raw count, you get something that is not random, but not very good. If you add sequence weighting, removing those 5 peptides that were very similar and was an artifact from how the data was sampled and not from how they are in nature, you get an improvement. If you add pseudo counts, this idea of extrapolating from the few observations to the whole possible peptide space, you see a further improvement. Do you add in this idea that position 2 and 9 are the ones having the most information, then increasing the information content at those positions by just multiplying them by a factor, you get a further improvement. And then you have, for 10 peptides, arrived at a model with this performance compared to a model trained on 212 peptides, 20 times as many data, which has a performance around 0.8. So of course you learn, you get a better model from more data, but you come a long way with small data volumes if you do these tricks of sequence weighting and pseudo count. So here we see that you have a lot of data. If you're lucky, you can construct an accurate motive, and these motives are very different. So this is for one particular MHC molecule, has a nomenclature AO201. This is the most frequently found across the globe in humans, but we all have different MHC molecules. That's why we are different, and why if you want to get an organ transplant, you have to find a donor match, because we have to match on these HLAs to have our immune system behave in a similar way between the recipient and the organ donor. And we're all different in that way. Each molecule has its own different binding preferences, and we can characterize those if we have data applying the techniques from today. And then once we have done that, we have all our motifs at hand in our computer. Now we can screen the genome of SARS-CoV. We can find the peptides that bind to my receptors or bind to all of these receptors, so we can make a vaccine that works in a population with different MHC molecules. And then we can use that in a vaccine to induce an immune response and hopefully save the patients. And there's actually ongoing research that people are applying these techniques to do this, and they are developing both within COVID, but also in cancer research, in developing cancer vaccines based on this exact thing. That was finding the binding motive of the MHC molecules, using those to screen the tumor genome to find peptides that can be used in a cancer vaccine. So it's I think it's pretty neat. So again, to summarize that, this is the essence of bioinformatics, right? You want to learn the motive from data, make a model of that, a weight matrix in our case, and then use that to screen. And then we can do that in this case for influenza virus, but also for SARS-CoV or for cancer, and pick out peptides with a high likelihood of binding to our receptor without doing any measurements. It's all computer. And in the end of the day, it turns out that these MHC binding peptides can now be predicted as accurate as you can measure them. So let's just stop here and say that you can use these logos to visualize binding motifs, and you can make a weight matrix even from a small number of data if you use these two techniques of sequence weighting and pseudo count. So that's actually the first presentation I have today, and I think we all deserve a break now for 15 minutes. So 5 past, we meet again. Are there any questions before we close this part of the session?
Por mi parte no, profe.
Bárbaro, entonces cortamos ahora y nos encontramos de vuelta 5. No sé qué hora es en Argentina, estoy muy perdido ahora. Yo estoy en Dinamarca, pero a las 5 past. ¿Sí?
Ok.
Chao, chao.
Gracias.